Said, welcome to the last Benbach for the season, to the bitter end, right? Not really. We have a really good program for you today. First, I'll actually introduce our intro speaker, Thea. So Thea did her undergrad in both computer science and biology at UBC, and then worked in Paul Plavides' lab as a web programmer for how long? A couple of years before she joined Fiona Brinkman's lab at SFU as a graduate student. And her research now is looking at metagenomics and microbiomes, and specifically looking at the effect of pollutions on microbiomes in environmental samples and identify biomarkers that can help the identification of contaminants and so on.
Great, thanks, Will. So as Will was just mentioning, my research looks at how contamination affects bacterial communities. One of the projects I'm involved in, we're looking at some bacterial communities that are collected upstream and downstream of agricultural contamination and comparing those two communities. And what happened was when I was looking at the analysis of this data, I came across some interesting issues around the normalization of metagenomic data. And so I thought, since this is a bioinformatics and metagenomics audience, this might be of interest to you guys. And so I'm going to talk about that today in my talk, Don't Push the Big Red Button, Normalization Pitfalls in Metagenomic Analysis Pipelines. So let's start at the beginning, as we always must. What is metagenomics? It is the study of genetic material recovered directly from a sample. In our case, we're looking at river water. So we take a sample of river water, we filter out the water, keep those bacterial-sized cells, break them open, extract the DNA, fragment the DNA, toss it on a sequencer, in our case Illumina, sequence the heck out of it, and then we get a set of reads per sample. Now, we analyze those reads. One of the reasons that metagenomics has been able to explode in popularity the way it has in the last decade is that there are a lot of tools available to analyze metagenomic data, some of which allow for very, very easy analysis of this data. For example, some web servers like EBI, Metagenomics, and MGRAST allow you to just take your FASTQ reads, upload them to a server, hit the big red Analyze button, and out pops gene family profiles and taxonomic profiles. And I'll mainly be talking about gene family profiles today. So let's get into this a little bit. What happens when we press that big red button? Well, we start with some QC. Can you see that? Yeah, okay. Some QC of the reads. Then those QC'd reads are used as queries for homology search against a database of proteins with annotated functions. Based on those hits, reads are assigned to gene families, and then those assignments are tabulated so that we get a table of gene family abundances per sample. Then we normalize those gene family abundances across samples in order to perform differential abundance analysis of those gene families in order to look for biomarkers of different sets of samples. And there are a number of ways to do all of these different steps and a number of criticisms of the ways that people do all of these different steps. But today I'm really just going to focus on this normalization step. So to talk about normalization, let's first look at the data before we normalize. This is a plot representing a gene table of abundances as a bar chart. So on the left here, we have pristine samples. So these are samples collected from an area that is not impacted by agricultural activity, and each column is one sample across time, sampled monthly over a year. And on the right, we have samples that were affected by agricultural activity. The height of each bar represents the number of reads that belong to this sample, and the colors represent reads that were assigned to a gene family. The gray represent reads that were not assigned to a gene family. And the first thing you might notice is that these columns have different heights, right? And this is not a surprise to any of you who've used multiplex data. From a sequencer, you're going to get different read numbers for samples just based on technical variation. So we don't want to consider that variation when we're comparing gene family abundances. So we need to normalize that variation away. The most popular, one of the most popular ways to do this is to normalize by percentage of reads assigned. And this is done in a lot of these push-button analysis software sets and done in a lot of really otherwise very high-quality tools. This is either the default or the only way you can get data out of the tool. And what you'll see is when we normalize by percentage of reads assigned, that is expressing the abundance of a gene family out of all of the reads assigned to any gene family, we actually have quite a consistent profile of these gene families across time and in the pristine versus agricultural samples. And this is something that's been seen in a lot of metagenomic studies where gene family profiles don't vary very much. But you may be wondering, what about these reads over here? What about those reads that were not assigned to any family? By ignoring those reads, we are ignoring, in some cases, 50% of the data. So if we consider those reads instead of ignoring them, and instead normalize by the total number of reads, not the percent reads assigned, what we see can actually look a little different. In the pristine samples, we see that there's not very much variation across time in these samples based on the percentage of reads assigned. But in this agricultural watershed, where samples are affected by agricultural activity, we actually do see quite a bit of variation in the percentage of reads assigned. So why is this? Well, there are technical reasons why you could have reads being assigned or not, maybe read length or read quality. But one of the big reasons that you can have a difference in the percentage of reads assigned between different bacterial communities is actually a change in the communities, or a difference in the communities themselves. So if you have a community of bacteria that are really well characterized, you have a lot of reference sequences in the database, you're going to get a lot of those reads hitting to sequences that are in the database. You're going to get a high percentage of reads assigned. If, on the other hand, you're looking at a microbial community that is more exotic, that has not had a lot of sequencing done for strains in those communities, then you're actually going to get much fewer of those reads being assigned to gene families. And so this can be actually a biological relationship. And further support for this being a biological relationship is seen in this data if we look at the agricultural samples here. You see there's sort of a trend in the percentage of reads that are assigned. And it turns out that this is correlated with rainfall in this watershed. And the reason that this is important is that we are looking at an agricultural watershed. So with increased rainfall, we get increased runoff, which means more of the contaminants, like nutrients and chemicals that are put on these fields, actually get transported into the water which we are sampling. So it really looks like there's a biological relationship here that we would be masking if we were to normalize by percentage of reads assigned. So point number one, if you normalize by percentage of reads assigned, you may be masking a biological relationship in your data. Okay, now normalization challenge number two. How do we get a meaningful abundance from our data? So when we talk about those gene abundances, often the way that gene abundances are expressed is out of a number of base pairs. So let's look at a simple example to see why this can be problematic. Let's assume we have two samples, sample 1 and sample 2, and they represent the same number of base pairs. If we look at a universal single copy gene, like gene A, you'll see that it has the same abundance per genome, which is represented by these circles, as a universal single copy gene would. However, if we just looked at the abundance of A per base pair in sample 1 versus sample 2, it would look like a really good marker between sample 1 and sample 2, even though it is biologically completely uninteresting because it's a universal single copy gene. On the other hand, if we looked at gene B, which looks like it might be a nice marker between sample 1 and sample 2, normalizing by or expressing abundance by base pair makes it look like gene B has the same abundance in these different samples because of this different genome size. So is this actually something that happens in real data? Yes, yes it is. In my data, for example, we had a difference of about between 2 megabase pairs and 4 megabase pairs, average genome size. And this can really affect what you're getting out as differential abundance of genes. So what do we do about this? We normalize, my favorite word, by the average genome size in this case, and that will give us counts per cell instead of per base pair, which will give us a same abundance for gene A and a different abundance for gene B. And I'm just giving you a brief overview here of these topics. So for more, I encourage you to go check out these papers. I'll have references at the end as well. So will normalization strategy change your results? Sometimes. In my experience, it has. So we looked at these samples that were collected upstream and downstream of agricultural activity, and we compared those samples that were most agriculturally affected versus those that were less agriculturally affected. And we found that depending on the normalization strategy we used, we got different gene families being differentially abundant. So what is the best normalization strategy? As always, it depends. It depends on your study goals, and it depends on your microbial community. If you have a large variation in the percentage of reads assigned in your community, it's going to be quite important that you consider that when you normalize. And the same goes for mean genome size. If you're looking for biomarkers and you are not interested in them having a biological significance, maybe mean genome size is not such a big deal for you. So again, this is a very quick overview. For more on this, I encourage you to check out these papers, and I'll have these slides will be up on SlideShare and they'll be on YouTube so you can get these references later. So finally, lots of resources have made metagenomic analysis very easy, but as ever, thoughtful use of tools is key to meaningful analysis. So for those bioinformaticians in the audience, hopefully this is another example of where you can say, aha, I am still very useful. And for those metagenomics people in the audience, hopefully this is an example of something you need to look out for in your data and a highlight of some papers to go look for more. Finally, I'd like to acknowledge my mentor and my supervisor, Fiona Brinkman, my metagenomics partner in crime in the Brinkman Lab, Mike Peabody, the rest of the Brinkman Lab, and the Watershed Project co-workers for the data presented today. Thank you. So I think we have time for a couple quick questions if there are any. Otherwise, I'll be at beer for sure. Yes?
So when you say that you normalize by genome size, is it like a constant or from the assignment?
So the genome size is going to be an average, and it's going to be a mean, really. So we're going to, the way that genome size typically is calculated is that you take all of your base pairs, you look for all of your universal single copy genes, whether that's one or a set of them, and then divide, right? And so you get an average genome size that you can use.
That community.
For that community, yes. And you're going to have a different average genome size if you have a different biological community. Okay, thank you.